huge libraries of drug compounds may hold potential treatments for a variety of diseases such as cancer or heart disease. Ideally, scientists would like to experimentally test each of these compounds against all possible targets, but doing that kind of screen is prohibitively time consuming. In recent years, researchers have begun using computational methods to screen those libraries in hopes of speeding up drug discovery. However, many of those methods also take a long time, as most of them calculate each target protein's three-dimensional structure from its amino acid sequence, then use those structures to predict which drug molecules it will interact with. Researchers at MIT and Tufts University have now devised an alternative computational approach based on a type of artificial intelligence algorithm known as a large language model. These models, one well-known example is ChatGPT, can analyze huge amounts of text and figure out which words, or in this case amino acids, are most likely to appear together. The new model, known as Complex, can match target proteins with potential drug molecules without having to perform the computationally intensive step of calculating the molecules' structures. Using this method, the researchers can screen more than 100 million compounds in a single day, much more than any existing model. This work addresses the need for efficient and accurate in silico screening of potential drug candidates, and the scalability of the model enables large-scale screens for assessing off-target effects, drug repurposing, and determining the impact of mutations on drug binding. Their research paper appears in the journal Proceedings of the National Academy of Sciences. In addition to the paper, the researchers have made their model available online for other scientists to use. In recent years, computational scientists have made great advances in developing models that can predict the structures of proteins based on their amino acid sequences. However, using these models to predict how a large library of potential drugs might interact with a cancerous protein, for example, has proven challenging, mainly because calculating the three-dimensional structures of the proteins requires a great deal of time and computing power. An additional obstacle is that these kinds of models don't have a good track record for eliminating compounds known as decoys, which are very similar to a successful drug but don't actually interact well with the target. One of the long-standing challenges in the field has been that these methods are fragile. Researchers have designed models that can overcome this kind of fragility, but they are usually tailored to just one class of drug molecules, and they aren't well suited to large-scale screens because the computations take too long. The MIT team decided to take an alternative approach based on a protein model they first developed in the year 2019. Working with a database of more than 20,000 proteins, the language model encodes this information into meaningful numerical representations of each amino acid sequence that capture associations between sequence and structure. With these language models, even proteins that have very different sequences but potentially have similar structures or similar functions can be represented in a similar way in this language space, and the researchers are able to take advantage of that to make their predictions. In their new study, the researchers applied the protein model to the task of figuring out which protein sequences will interact with specific drug molecules, both of which have numerical representations that are transformed into a common shared space by a neural network. They trained the network on known protein-drug interactions, which allowed it to learn to associate specific features of the proteins with drug binding ability without having to calculate the 3D structure of any of the molecules. With these high-quality numerical representations, the model can short-circuit the atomic representation entirely, and from these numbers predict whether or not this drug will bind. Another advantage of this approach is that it takes into account the flexibility of protein structures, which can be wiggly and take on slightly different shapes when interacting with a drug molecule. To make their model less likely to be fooled by decoy drug molecules, the researchers also incorporated a training stage based on the concept of contrastive learning. Under this approach, the researchers give the model examples of real drugs and imposters and teach it to distinguish between them. The researchers then tested their model by screening a library of about 4,700 candidate drug molecules for their ability to bind to a set of 51 enzymes known as protein kinases. From the top hits, the researchers chose 19 drug protein pairs to test experimentally. The experiments revealed that the 19 hits, 12 had strong binding affinity in the nanomolar range, whereas nearly all of the many other possible drug protein pairs would have no affinity. 4 of these pairs bound with extremely high sub-nanomolar affinity, so strong that a tiny drug concentration on the order of parts per billion will inhibit the protein. While the researchers focused mainly on screening small molecule drugs in this study, they are now working on applying this approach to other types of drugs such as therapeutic antibodies. This kind of modeling could also prove useful for running toxicity screens of potential drug compounds to make sure they don't have any unwanted side effects before testing them in animal models. Part of the reason why drug discovery is so expensive is because it has high failure rates. The researchers say that if they can reduce those failure rates by saying upfront that this drug is not likely to work out, that could go a long way in lowering the cost of drug discovery.